Okay, good morning, everyone. Thank you for coming on this warm day for the one of the few last seminar, the one before the last seminar of the year. I hope people online can also hear me. If you can't or there's any problem, let us know in the chat. So today we have the pleasure to have Ludovic Orlando. Unfortunately, he couldn't be able to join us because of trains not working in France on 4 days. And before I give the speech to Alouen to introduce Orlando and Ludovic, sorry, I want only to say that maybe you have received the call for the next round of speakers is still on. Please let us know if you send us a message if you have somebody in mind to invite for the next year. And the last talk of the last seminar of the year is next Friday, and then we come back. We reconvene in September. Okay, the floor is yours.
Thank you. Thank you for coming. I will just make a very short introduction of Ludovic. Ludovic, you may have may have heard of him. He's a biologist working in Toulouse. He's the director of the Center of Microbiology and Genomics in Toulouse. And he's one of the researchers pushing the limits of ancient DNA analysis, sequencing more and more ancient specimens and more specimens overall, and exploring many aspects of ancient DNA and ancient DNA preservation, including, for example, epigenetic, which not only people are exploring this field. So Ludovic is well known for his work on horse domestication, and is currently running a synergy grant looking at the makeup of the modern horses. And finally, since December last year, he has been nominated at the French Academy of Science. So he's a big French name. So thank you, Ludovic.
Thank you so much. I believe you hear me at the moment. Yes.
Yes.
Great, amazing. So thank you so much for hosting me today. I'm really disappointed to not be in person in Montpellier, because I would have loved, of course, to interact a lot more with you and present the kind of the work we've been doing over the last, say, 15 years now, working on horses and using genomics to understand what is it we did with them over the last, say, 5,000 years. So I'm going to start just reminding you that there are not so many horses living on the planet today. In fact, it's 60 million. That means that there are more French people than horses on the planet. And they are mostly known for being a horse racetrack champions. And typically, when we hear about them, this is really for their very important racing results, but also for funny things going on during racing. But over the past, in fact, horses were a lot more numerous, and they were a lot more sharing their lives with us. They gave us the speed in the first time of history. So that means that with them, we've been able to travel a lot faster across the world, meaning that any part of the world was pretty much connected to unprecedented level as long as we had domesticated the horse. So that means that places we could reach in months could be reached now in weeks, or places we could reach in weeks could be reached in days. And that, of course, totally changed the pace of human society interactions across the world. So oftentimes I say that horses have really, truly globalized the world for the first time, making it modern to some extent in terms of fluidity of exchanges at the geographic level. But of course, horses also gave us a way to kill each other a lot more efficiently. And so that revolutionized warfare. Think, for example, about the past. Maybe you will picture Genghis Khan somewhere in your mind, and there will be no Genghis Khan without horses, basically. Likewise, there will be no Alexander the Great defeating Darius III here on this mosaic. There will be none of them without horses, because they were such horse masters. So I bet that if I ask you now to think about our past and human history, soon enough you have something in mind, and soon enough you will see that there are horses behind us pretty much during all of our history. And surprisingly, perhaps, given how important they were for us during history, they were really basic questions that were left unknown when we started these projects. So basically, questions such as when did we domesticate the horse for the first time, as well as where did we do that, and how transforming the wild animal into a domesticate, meaning some kind of companion for us, all those questions were left unknown. And so this was not for the lack of trying. In fact, scholars have been trying to solve those questions for more than a century, but largely failed. So we decided to really leverage genomics to sort of answer those questions. And the kind of genomics we do is that not only do we sequence horses across the world, living horses across the world, to really assess and understand their geographic structure today, but we also do that in the past, leveraging the survival of ancient DNA molecules in archaeological remains such as teeth and bones, basically. And so the reason why we decided to do ancient genomics as well as modern genomics, in fact, is that genes and genomes, in fact, tell you a lot about individuals. What is it they look like? Because there are traits we can predict genetically because we understand so well the genotype to phenotype association. But of course, DNA is shared across genealogies. So that means that DNA gives you a sort of information at the family and population level. And so if typically we can travel back in time, where today will be on top of the screen here, and the past will be in before the Common Era, BCE, some point back in time, we could actually reconstruct patterns of population structure as a dynamic target through time. And that maybe will tell us something about where everything started and how it unfolded space-wise across time. Of course, if you have complete genome time series, that means that for each and every mutation across the genome, so time will be x-axis here, and the allelic frequency of your favorite allele across the genome will be the y-axis. Well, you could track when allelic frequency started to rise much faster than by drift. So that means that with ancient genomic time series, virtually for every single allele across the genome, you will be able to measure artificial selection and start to appreciate when artificial selection started, but also measure really the selection coefficient that is associated to sort of describe the strength of selection back in the days. So this is really what we wanted to do. And I should stop at that stage and really insist on one thing that I believe you should remember from this seminar, is that our work over the last, say, 15 years has shown something very simple, is that the impact of the domestication was tremendous on horse genetics. So this is a PCA that summarizes the genetic diversity that we discovered at the genome-wide level by sequencing more than a couple of thousands of ancient horse genomes spread across the last, say, 50,000 years. Each point here is an individual genome. And the farther where they are 2 points, the more diverse the genetic diversity is. And so this is the diversity that existed before domestication. And if I do the same plot at the same scale after domestication, this is what is left, basically. So clearly, the take home is that the domestication of horses had a dramatic impact on the extent of genetic diversity. Clearly, we seem to have selected or bred one particular genetic type that we have successfully spread out across the world. So now I'm going to try to address with you those three basic questions: where, when, and how did we do this massive reduction of genetic diversity? So starting with time. So you have on this particular slide on the x-axis the time. Negative means before the Common Era. So when it's 0 is 2,000 years ago. When it's -10 is 12,000 years ago. And I just represent the genetic ancestries in two colors here. The two orange, these are the modern domestic horses. typically the kind of ancestry you find across the world today in modern domesticated horses. And black will be all the other kinds of genetic ancestries that we haven't covered. I show them in black. And there is a very clear temporal pattern in this particular plot. So if I zoom in the most terminal part of the plot, you realize that basically the modern genetic type of domesticates sort of emerged at the second half of the third millennium BCE, which means 4 to 4.5 thousand years ago, basically. It did not exist before, and so that means that horse domestication, the modern genetic type of horses, emerged very late in human history, 4 to 4.5 thousand years ago, end of the third millennium BCE. And at the same time, there is a striking pattern as well, which is that at the minute that the modern domestic horse lineage emerges in orange, you have all the other types of genetic types that sort of vanish, right? The black points are less and less frequent on the same temporal axis. So we could view domestication as the success of the orange type that I show here, but we could equally think about domestication as the loss of all the other types that were present in my previous slide. So if I look at now the genome data we have to reconstruct effective population size variation, precisely in the third millennium BCE, the moment when the modern domestic type emerges genetically, we recover a very strong signal of a bottleneck, which is associated with domestication. Anywhere between, say, 2700 BCE to about 2100 BCE, there is a population collapse of horses down to a couple of hundreds of reproducing animals on average, basically. So that shows you that domestication, the bottleneck, was dramatic. It was several orders of magnitude large bottleneck. But at the same time, it lasted for about, say, 500 years. So the domestication bottleneck spreading across 500 years or 400 years is a sort of time estimate of the time it took for us humans to generate the genetic type of modern domesticates. And then once that was done, basically horse populations expanded dramatically, exponentially in almost a blink, basically. Within, say, 50 years, the population went from only the founders to many dozens of thousands, basically. This is the moment where we started to have control the horse reproduction so well that we could replicate them and produce them in large numbers to actually move around the world, basically. So that solved the when question. Basically, the when is surprisingly recent. This is something that took place 4 to 4.5 thousand years ago, end of the third millennium BCE. So that means that horses are one of the latest herbivores that we have domesticated. Even the donkey was domesticated 3,000 years earlier, somewhere in East Africa. So now I'm going to target the where question. Where is it that we did so? So I'm projecting here the genetic ancestry blends across Eurasia before the third millennium BCE, so before 3000 BCE. So that means before the domestication, to sort of learn what was the population structure of wild horse lineages before domestication. And you see that you have different colors segregating in different regions, suggesting that probably horse populations, when they are wild, they don't move around too much. Otherwise, genetic ancestries would be shared at a very large distance. And this is not what you see. So we could use those patterns to sort of measure the migration rate, as shown here with the continuous ribbons. And when those ribbons are brown, dark brown like here, it suggests negative migration rates, meaning no migration whatsoever. So you see that Eurasia, before domestication, from the horse perspective, can be largely thought as a place where horses do not move around. Horses, when they are wild, it seems they are born, they live, they reproduce, and they die pretty much at the same place. So that means that genetic lineages are sort of isolated by distance, basically. So what happens in the third millennium? Well, not much. This is the same kind of patterns that you see, you know, brown negative migration rates, meaning no migration whatsoever. But if you look at what happens right after the third millennium, after the horse was domesticated, and right at the time when the demographic history exploded, you realize that the picture changes dramatically. Because right now, there is only one genetic type, only one blend of genetic ancestry, the one shown in orange here, that is actually spread across the whole of Eurasia. So it means that this is this particular genetic type that was domesticated during the domestication bottleneck that was reproduced in large numbers and so spread by humans together with them as they migrated around. So, of course, the question we had when we saw that, of course, it's a massive impact of us on the population structure of horses. But the question we wanted to answer is where from, really, because that will show the origin points of horse domestication. So to answer that particular question of where from, we just trained deep neural networks on the genetic population structure that existed prior to domestication. So say prior to the domestication bottleneck. So you take all the genomes you have, they are geolocated and temporally dated by radiocarbon dating, and you learn with DNNs what are the genetic motifs that are structuring the ancestry distribution at the spatial level. Once you have that, you can take any of the genomes you have after that moment, once the genetic type of modern domesticated horses emerged, the orange one, and you could ask for each and every of them, where were their ancestors living? And for all of them, the thousands we have sequenced from 4,000 years ago to today, we had all the time the same answer, which is the pseudo likelihood surface of all the predicted locations of where the ancestors were living, which solves the where question. It seems that horses were domesticated by the second half of the third millennium BCE in this specific region that we call the southwestern Russian plains today, which could be thought as the lower Don Volga region today, a tiny, tiny region, yet that had massive global consequences after the horse emerged from there. So that's all the where. So I'm left with the how, but before going to the how, I'm going to illustrate something about the mobility, because right after horses were domesticated there, we see in our data a massive migration of that genetic type outside of the domestication homeland or center, if you will. So we can see that many ways. So here I just measure isolation by distance pattern as a function of time. Time here is in BCE. So what you see here in bluish or gray, this is the moment of the third millennium BCE, the moment of domestication. You realize that before the domestication, horse populations were isolated by distance massively, so no mobility too much. But after they were domesticated, basically there is no isolation by distance anymore, meaning that horses genetically living close to each other or living 10,000s of kilometers away, they are the same genetic type, no isolation by distance anymore. So we could view it in a different way when we look at our data. So here I'm showing the earliest domestic horse that we have in our data set. It lived in the north of the Caucasian range in the southwestern Russian plains, as I've told before. And this is dated to 2200 to 2400 BCE. So now I'm going to do the same for the next century. Where were they living? Oh, you see that within a century, we see the domestic type already in Armenia or Georgia, as well as in Anatolia, Turkey today. If I do that for the next century, 2000 to 2100, you see that horse domesticates are already in Europe as well as in the Ural Mountain. And if I move forward in time, you realize that it takes no more than 250 years, in fact, for horses to be already in Iberia, Spain, or Portugal. So it means that within 250 years, that genetic type of horses that was domesticated in the steppe of Western Russia actually could travel across all of Europe and actually reach out to also the Altai on the Asian range. And what is really cool about this pattern is that it matches what archaeologists have mapped as well. For example, archaeologists, they can map not genes, but in fact, they can map all all the equipment that they can excavate that are related to horse control. So for example, here you see some cheek pieces that typically you put in the mouth of the horses to control their movements. And you realize that there is a massive expansion of those cheek pieces right at the beginning of the second millennium, between 1900 and 1700, which means that not only the horse expands of a new genetic type, the modern domestic type, but also all the instruments for controlling their movements. And that's the same for chariots, because we could prove that the distribution of chariots with spoke wheels actually matched also the time and spatial expansion of this domestic type. So now I'm left with the how question. How come did we manage to transform wild animals into some animals that were so useful to us? And so as a geneticist, one easy way to try to tackle that question is to say, okay, I bin all the horses that we have domesticated in one category, and genetically, I'm going to compare that category with all the other ones we never domesticated, right? So I make two groups and I FST contrast every single SNP across the genome to sort of identify the mutations that segregate at high frequency in the domesticates, but remain at low frequency in the wild animals. That will actually identify a series of candidates where the frequency of one mutation was selected positively, so much so that it raised really large in high frequencies in the domesticates, but never did so in the wild. So when we do this FST contrast genome-wide, you have 31 autosomes in the horse genome. You realize that there are two loci that stand out, one on chromosome 3 clearly and one on chromosome 9 here. So I'm going to start with the one on chromosome 3. We could show that the allelic trajectory for the alternate allele was really like this one. So time is on the x-axis, and I show T1, T2 is somewhere around the domestication bottleneck. So you see that before the domestication bottleneck, there was small allelic frequencies. During the domestication bottleneck, that mutation came in high frequency and remains so ever since, right? So when you have this allelic frequency, you can actually measure selection associated with it under a neutral model, which we did. And we identified that it was a sort of selection coefficient of about 3 to 4%. So quite significant selection. So you want maybe to know what that particular locus does, basically. So the mutation is located within the ZFPM1 gene on the chromosome 3. And typically, mice knockouts and knock-ins have been done before. So we just leveraged the literature to understand what was going on. And people modified the ZFPM1 by knocking out or knocking in that particular locus. And they typically tested the mice under these frameworks. So you have a platform with places where mice will be protected behind walls or will be left in the open here in this platform. And the camera is measuring the time that the mice spend on the open or under the protection of the walls as a sort of measure of how aggressive or anxious the mice are. So it's typically a sort of anxiety-based phenotyping that do. And you see that you are male or a female, a mouse, it doesn't matter if your ZFPM1 gene is knocked out. Basically, you spend a lot less time in the open, meaning that you're a lot more anxious, in fact. So clearly, we can view ZFPM1 as a locus that is a controller, a switcher of behavior from anxious to less anxious. So maybe we made the hypothesis that this selection we saw really early in the domestication bottleneck was actually a behavioral selection for more docile, less anxious kind of animal, which will help us interact with them. But the most interesting for us was the second one associated with highest FST values that was located on chromosome 9. And in fact, the FST peak is located in the promoter region of a gene that we call JAS2 C GSDMC. And for that one, actually, we didn't know much. So we had to do all the functional work and validation. So for example, if you look at the allelic frequency, this is right expanding at the domestication bottleneck, no earlier, and with selection coefficient of at least 10 to 15%. So it's a massive selection that goes on that locus, right? So what we did to sort of functionally understand what was going on is that we started to measure phenotypes in living population of horses. So for example, phenotypes such as the body length to height ratio, which is a measure of the shape of the horse body, basically. Why that one is because it's related to racing and to mobility. So we thought that maybe this selection was associated with a mobility trait. And if you look at population in Ningxiang in China, where we could find actually enough variants of two genotypes, there is a significant association of the genotype those horses carry with the morphotype, basically, with the body length to height ratio. And we could do that in a global population and also retrieve a significant phenotype to genotype association. So there is clearly a phenotype that we could see visually in horses, which was encouraging for us to sort of dig for further, right? So I should say at that stage that the variant is located here in the center, but it's the promoter of GSDMC, but it's also flanked by two L1 insertions, and that those three variants are actually very highly linked. They are in very strong linkage disequilibrium. So you could think about that as a locus of about, say, 250 kb that comes and segregate as a block in the modern domesticated horses. So we then try to look for molecular phenotypes. And for example, if you measure transcription level of GSDMC in the blood of horses that carry any of the variants located in that block, you see that the homozygous derived variant here is associated with an underexpression of GSDMC. We could also show this in transitory expression assay, transfection assays in different cell types. So clearly there is not only a phenotype we could see with our eyes, the morphotype, but there is also a molecular phenotype under-regulating the transcription of GSDMC. So that was very encouraging. So that's why at that stage we thought we should switch to a sort of model organism to try to dissect or understand what was the phenotypes that those variants could drive. And so we opted for preparing some mice model because they can easily be manipulated in the lab as opposed to horses. And we prepared a KO for GSDMC, which is a KO where exon 12 of GSDMC is shortened, and we could prove that this shortening of exon 12 is behaving as a downregulation of the transcription of GSDMC. So it's a sort of model that is analog to the molecular phenotype I described just earlier in horses. And we looked at morphotypes, and we looked at mobility traits. So I'm going to show here one morphotype by measuring the height to length ratio of the spine. Here you see the spine under CT, basically. And you see that if you have a KO mouse, basically there is a significant reduction of the height to length ratio of the spine, which is called the kyphosis index. In other words, if you underexpress a GSDMC, as we see in the horse with the selected variants, you have a flatter back, basically. So the back anatomy changes and it becomes flatter. And we could explain the ultrastructural variation changes that happens to make the back flatter. So that's one phenotype very interesting for horses. The back becomes flatter. Where do we sit on the horse? On the back. So maybe we are on something quite interesting here. So the second thing we did with the phenotypes was to measure some kind of locomotory phenotype. So for example, we can measure the time it takes for those mice to remain hanging on this wire here. So basically, the time it takes for them to fold is a signature of how strong the forelimbs are. We could also train mice to walk on a rotating device and still measure the time it takes for them to fall. We call that the latency to fall. And this latency to fall is a direct measure, quantitative phenotype for motor coordination. And so no matter which assay we do, we could demonstrate that the KO mice, those that underexpress GSDMC, as in the horse phenotype, clearly are associated with a stronger forelimb strength, but also with an enhanced motor coordination. So that's why we think that we have a motor coordination phenotype as well as strength-related phenotype associated with the domestication of the horse. And what is really cool for us is that there were GWAS studies done in humans that also show that this time overexpression of GSDMC variants are actually associated very significantly with a syndrome that we call the lumbar spinal stenosis, which is a syndrome in humans where people overexpressing GSDMC are actually not able to move anymore. They have a change in the back anatomy that makes walking even really nasty and difficult. So clearly, because we see a selection of those variants right at the time of the domestication bottleneck, because the phenotypes are mobility related and motor coordination related, and because they are massively selected right at the time before horse moves across the landscape, we think that we have identified some of the loci that made this possible horseback riding even possible during history. So I'm going to spend the time that I have left, maybe the last 5 or 10 minutes, to talk about what happened since. And since I will start with selection. So the same way we have allelic trajectories for those two variants, actually we could apply allelic trajectories or draw allelic trajectories more so for every single position across the genome. So we looked at 266 of them because their association with the phenotypes were really well understood. So here you see the path from today to the past in generations ago. The domestication bottleneck is gray here, and anytime you have a column, that's a locus for which we show empirically a shift in the selection coefficient at that locus. For example, here on TBX3, that's one of the genes we were interested in. You see that it was evolving neutrally before, but at that time, 350 generations ago, there was a shift to strong positive selection with selection coefficient of about 20%. So what is interesting about that gene is that we could show years ago doing GWAS in modern horses that this particular locus is significantly associated with a phenotype, which is the size at the withers of horses. So genotypes that you carry at that location condition very dramatically the size that the animal develop. And so we could actually demonstrate that this mutation was causal for the variation, again, using mice models. But what is interesting is to understand the allelic trajectory. So what we learn is that there was no selection until 370 generations ago, which translates about 728 BCE, so almost 2.7 thousand years ago. And at that stage, there was selection going on up to the present day with a shift in the selection coefficient somewhere around 220 generations ago, which translates into the end of the 4th century CE. So that means that size did not matter too much for horses until 2.7 thousand years ago, which means that from early domestication, 4.1 thousand years ago, to 2.7 thousand years ago, almost 1,500 years, size was not selected during horse evolution. So instead of describing for you everything related to selection, I thought I should finish by just showing you something about migration, because we could actually reconstruct migration back in time. So for example, I'm going to focus only on the Americas, which is a region that is very interesting for when it comes to horses, simply because our classical model predict that there used to be a horse population living in the America before in the Upper Paleolithic, but they all became extinct somewhere around 12,000 years ago. We think about this because we don't find any fossils anymore. So if that model is true, it means that it's the conquistadors who brought horses from Europe as they colonized the Americas for the first time. So horses came back with the colonization. And our models predict that basically horses remained in the hands of the colonizers for a while, but actually were seized by the indigenous people by the end of the 17th century. Precisely in 1680, which is the time of the Puebloan Revolt in New Mexico, which is a time of 20 years when the indigenous people actually took over the Spanish power in New Mexico. And I was contacted a few years ago by a Lakota scholar. She actually didn't believe in this model based on a lot of interviews she made with indigenous elders, basically knowledge keepers in many indigenous nations. So she actually published a PhD on this. So we thought, and she proposed a different model, that horses did not become extinct in the Americas, but actually when the conquistadors came in, there was a mixture of two horse types that was actually the source for the modern populations until the present day. So I replied to her that I will be thrilled to work with her on this very question. I did not know at that stage that the work we will develop together for 6 years in my lab will actually end up winning the Newcomb Cleveland Prize, which rewards the best paper published in science. It was in 2024, I believe. And this is what we've done. So we sequenced a lot of ancient horses from the Americas in red. They are historical, so from the past. We compare them in pink to the horses that exist today in America, specifically the horses of the indigenous nations, and we started finding interesting things. So, for example, there is a horse bone that was radiocarbon dated to before the Puebloan revolt, clearly in the posterior distribution of the radiocarbon date. But that particular bone came from a horse that was fed maize, basically. We could demonstrate that with the isotopes. So clearly maize at that stage was just a native plant. So clearly that horse was actually with indigenous people. It was fed by indigenous people, not Westerners, not Europeans. So that means clearly that native indigenous people had access to the horse before our classical model predicted. In that case, probably the end of the 16th century, not 17th century. But genetically, those horses, the one that I've just shown, or any of the horses that existed in the colonization time in red, or until the present day, including those living with the indigenous nations, none of them genetically is different from the modern domestic type. They are actually not carrying the ancestry of the horses that existed in the Americas in the Upper Paleolithic, in the late Pleistocene, meaning that they are very distinct types. So we could model the genetic type of the horses from the colonization time period and today as really European in ancestry. So I'm going to illustrate that with this biplot where you measure on the y-axis how Spanish genetically those horses are versus how British they are. So Spanish and British are the two colonization powers. You see that those horses that arrive early in the colonization process in red, they are more Spanish than British. Clearly, this is the direction of the colonization. But those that live today in America, they are a lot more British than they are Spanish. So clearly you see in the horse genetic landscape that there was a shift from Spanish ancestry to really a British ancestry. So I don't have the time, I think, to cover all this. So I'm going to jump to my conclusion immediately. So I hope that I convinced you that really when you will think about horses in the future, there's a lot more in their history than just racing. Actually, horses have told us a history of diversity that has deep historical roots, at least the late third millennium BC. And their history with us has been a history of both mobility, power, but I will argue as well mutual dependence, because the domestication process takes two partners accepting to coexist and to co-evolve together. And with that, I would love to thank my sponsors, specifically the ERC, to have supported me for almost the last 10 years now on this sort of projects. And of course, you all for the kind invitation and the kind attention. Thank you very much.
Thank you very much, Ludovic. So I invite you from here to speak loud so that Ludovic and everybody can hear the questions, and also people online to probably leave your questions on the chat. We'll also take the mic. Like, maybe we wait a few seconds. Have some go.
Yeah, I've stopped sharing just so that you will be able to see the chat actually.
Like if you need to do this button, no problem. So yes, any questions?
Thank you very much for your talk. I was wondering in terms of method, how do you do to reconstruct allele frequency trajectory over time?
Yeah, so very simple, in fact. So we sequence the whole genome. And so there are two ways. So the easy way is that you have sequenced the whole genome. So that means that even if you have limited coverage, you have randomly sampled through sequencing one of the two alleles at every single location across the genome, right? So if you have that from one genome, you know that at that position there's a A or there's a C, for example. Now you do it for many different genomes, thousands in fact, all of which will be radiocarbon dated, right? So that means that you have per time bin, say within time windows of say 250 years, you have the counts of the alleles in this time period, okay? So that means that you have the counts, which is an estimate of the allelic frequency, basically. So that's the brute force and simple way to do this. There is another way that is now very well working, is that instead of relying only on low coverage, which only give you information of one of the two chromosomes that are carried by a single individual, you could actually statistically impute the genotypes genome-wide, right? And so we can do that with high accuracy in horses because we have sequenced so many, both in the past and present, so that we could show that as long as the allelic frequency is larger than 5%, we have a 99.7% accuracy to statistically impute any genotype in the population. So this time you will have explicit genotypes, and so you will reconstruct allelic frequencies the same way as if it was a modern population. You have simply time to account for.
Thank you very much. Very clear. Thank you.
There's one, Arnaud, would you like to ask your question? Let's try.
Yes, do you hear me?
I do.
Okay, great talk, Ludovic, and great results. I like pretty much this the evolution of domestication in general. So I have a very I have a question which is exactly the opposite. My question is, what about fertilization of domesticated horses? Do we have now any evolutionary insight about this retro-domestication, if we can say so?
Yeah, so that's a really fascinating question. Thanks, Arnaud, for the kind words and also for the question. So this is a question that, of course, fascinates us as well. In other words, is domestication reversible, right? Genetically or maybe expression-wise, right? So I don't feel we have enough data yet to give you a very important insights, but I'm going to comment on results that I hope will be published fairly soon. So we have sequenced the genomes of a number of feral populations across the world. So they are not ancient. They are feral today. For example, there are populations that were actually abandoned on some islands, right? And we know the date when those horses were abandoned, and we know that ever since they have lived without us, right? So clearly there is one very famous in the Sable Islands, for example, which is Northeast Canada. And so we could track, after 200 years after they have evolved separate from humans, what happened to their population. And the same goes for other kind of populations in different Mediterranean islands and also in different islands in the Pacific, basically. And so what we looked at is the relaxation that we could see on the genome, right, basically. So what we saw is that depending on the case, there is a strong demographic impact on this. Depending on the founder population that was involved, you will have a starting situation that will include more or less deleterious variants. And depending on the demography, we see fast or slow purging of those deleterious variants, right? So we see clearly an effect on measures such as inbreeding and deleterious load on the genome, basically. And clearly, in general, I'm saying in general, because there are exceptions, founder populations of feral populations are small. And so because they are small, drift is more efficient than selection. So the purging of deleterious allele doesn't work as well. So clearly those populations that I'm referring to, their genomes is inflated in deleterious variants. But there are exceptions. One very important exception is the Mustangs from Northern America, typically because that population is very large demographically, right? Typically, if you think about Northern America, you will think about the cowboys and those Mustangs, basically. And so there we see that purging was very efficient. So there are no single rules. We will love to be doing the same project, but not looking at just negative selection and purging or inbreeding, but also looking at expression, right? meaning that all the genes that are overexpressed or downexpressed due to the domestication, all their expression profiles reverted as well. But so far, I've failed dramatically accessing those fresh tissue from those horses from living in feral, right, in feral conditions. And I'm not giving up because there are populations, Mustangs, or in America, in Australia as well, we call them the Brumbies, where people shoot from helicopters, hundreds of them every year to control the population. And so at some point, maybe we will be authorized to go, when they shoot, to go and sample different tissues of those animals, basically to really measure with RNA-seq the expression pathway. But so far, we have totally failed. I should add one thing about fertilization is that we showed with our work, I didn't have the time to cover that today, that typically what we call the Przewalski's horse, which was thought before our work as the last truly wild horse population living on the planet today, is in fact a feral population descending from an independent domestication process that started 1,000 years before the one that I commented today. So clearly, people domesticated first the horses in Central Asia, but then those people disappeared. We could prove that by sequencing the genomes of the people, in fact. So they disappeared. And so the horse was not controlled anymore, so went back feral. And it survived until the 19th century when it was rediscovered by geographers who explored the south of Mongolia. And they gave the name of Przewalski's horses to that population. If you look at this population, it's massively inbred because in the 20th century, very recently, in the last 100 years, the population has almost become extinct. It only survived in captivity in zoos. So it's not a great population for us to really study the fertilization process unless we travel back in time. So for that population, we travel back in time to try to address the dynamics of fertilization. But I cannot say we are close to understand that for the simple reason that when we sequence horses from the past in the last 4,000 years, oftentimes they are genetically domesticated, as I showed, right? So we cannot design a project where we could tell to the archaeologist, oh, find for us, say, 500 Przewalski's horses in the past, right? Because oftentimes the sites they excavate are sites with humans, and therefore they contain only truly domesticated horses. So for the moment, to give you an idea of numbers, we have now sequenced more than 4,000 ancient domestic horse genomes in my lab. We only have 10 ancient Przewalski's genome, right? So we are really decades away from being able to assess that empirically.
Thank you. Thank you very
What about Pauline? I'll just take one more online. Okay, Pauline, would you like to ask your question?
Yes, do you hear me?
Absolutely.
Okay, so thanks for the presentation. I just have a question about like the different population structures that you presented. We can see that on the map you have a very different structure in Spain.
Yeah.
Do you know why?
Yeah, very good spotted. I mean, that's really great that you saw that, Pauline. So she's referring to a map that I showed rapidly, the one, I guess, Pauline, before the 3000 BCE, right? Where and after 2000 BC, everything was orange and there was this sort of purple in Spain. This is what you refer to, I guess, right? So so that map that I've shown, and I'm sorry that I was not really clear about that, was any horse that was living from 2000 BC to 0 CE, right? So say the second millennium and the first millennium BC. If I want to translate that in years ago, that will be 4,000 years ago to 2,000 years ago, okay? So it's a pile of 2,000 years that I show. If I would give the date of the Spanish horses on this map, they are 3,900. Right. So they are actually living right at the transition between the third and the second millennium. Right. So they show still that they are purple, so wild in this region, simply because at that stage, the domestic horse did not yet reach out to Spain. In fact, the domestic horse reached out to Spain. We could prove that in a paper last summer in Nature Communications. It reached Spain, the domestic horse, in 1850 BC, 50 years later. Right. So if I showed the map, not 4,000 years to 0, but 1850 to 0, everything would have been orange, basically. Right.
Okay.
So, in other words, in Spain, because this is so far west in Europe, it took time for the orange type, the domestic type, to arrive from the steppe to this far and remote place, which is Western Europe, right? It took 250 years, right? It was not a sort of overnight replacement. In fact, in the paper that I'm referring to, the 2025 paper, we could show that they will coexist for a while, and the while being almost 1.5 thousand years, right? So you will have this purple ancestry surviving in Spain at low frequency until, say, 200 CE, 300 CE, okay? And then after the Punic War, they totally disappear. Sadly enough, that lineage became extinct eventually.
Okay, thank you.
So related question. So when did you find the last wild horse in Europe?
Yeah, so that's a good question, Alwyn. So I can certainly answer with non-published data. So we have a paper we are trying to finalize where we have identified a number of wild lineages surviving until very late, actually. For example, we published a few years ago some genomes too from what we call tarpans. And tarpans are typically the wild horse from the steppe, from the Russian steppe, okay, or Poland and Russia, Ukraine as well. So this one we knew was living until the late 19th century for the very reason that we have pictures of it, right? So there are people who have photographed it, right? End of the 19th century. But that was the exception, we thought, right? Because we only knew about that one. But in fact, now in our extended data set, we found that in the Middle Ages, for example, in Germany, we find the equivalent of the wild type still in the middle of the Middle Age. So it seems that in pockets of populations, the wild animals survive until very recently. Maybe in some places until the 19th century, maybe in other places 200 BCE, as I've just said for Spain. In Germany, it seems that it's until the middle of the Middle Age. But what is very true in our data is that they eventually all go away, right? They all go away. So we should think about the replacement extinction process as a very dynamic process, both in space and time. There are regions when it took a long time to replace and lead the wild animals to extinction. For example, the Russian steppe, it took until the end of the 19th century, but there are places where it took a lot less time. For example, Spain, it took 2,000 years less, for example, okay? And so then the question becomes, why do we have so many domesticates in our data set and so few wild, right? And I guess archaeologists will realize immediately that what they excavate in general are sites where there is human activity, right? Typically settlements where humans are buried, or tombs or sepulchres and things like that. And oftentimes when there is humans involved, well, it's the domesticates that will be overrepresented, right? So you don't see archaeologists going wild and randomly excavate places with no sign that there will be something to even discover. So I think there is a strong bias, in other words, in our data set where we tend to overrepresent the domesticates because we tend to look at places that are associated with humans. So, but at the end of the day, it remains very true that if you sequence thousands, as we have now, modern horses across the world, including in remote places, I mean, places where there is no horse industry, village horses, you don't see any of the wild types anymore. The only ones you discover are what we call the Przewalskis or the orange type, the modern domesticated type. So eventually, they all became extinct. And we don't think they were, hunted to death. We think simply that the sheer number of expanding domesticates, we produced horses by hundreds of millions, right, was simply enough to extirpate the wild populations in small and smaller pockets, right? So they could not really thrive in any environment in the end.
Can we take one more from here and then online?
I have a question about the future of Przewalski horses, because they suffered from a huge bottleneck, remained 20 horses.
Yeah.
And then they have been reintroduced in the wild. And I wonder whether species will suffer from a very high inbreeding depression. So is it realistic to think that this species will survive? And will it be possible to try to reinforce this new population by crossing them with other horses? I don't know whether Przewalski horses are different subspecies or species.
Yeah.
What is the possibility to cross with other horses?
Yeah, brilliant question, and very important, in fact, because we're talking about the survival of the other type, the only one that exists still until the present day, right? So I should start saying that Przewalski's horses, the bottleneck that took place in the 20th century is down to 12 to 15 founders. It's 10, 12 to 15. So indeed, they survived something dramatic, right? 12 to 15 founders. Today, there are 3,000 of them around the world, but they all descend from those 12 to 15 captive founders, not much. They survived, yes, but in captivity, right? Really because they were protected. And we could show that they were protected, but some of them were actually even crossed in zoos with the domesticates. In fact, there is a very popular lineage in the pedigree of the Przewalskis that is very popular numbers-wise, that is really admixed with modern domesticates genetically. So in fact, there are no Przewalski's horses today that are really, say, pure ancestry-wise, right? Sadly enough, the bottleneck was such that they have some blocks of the genomes of small size that come from domesticates. But I would argue it's definitely worth preserving them. Of course, they are wild, or they are, I would say more feral if you ask me, but they are a population that shrinks, so we should preserve that. And that's also the descent of the first domestication process, so there is another added value to preserve them. They are sort of living fossil of the earliest domestication. But what are the chances? So I should say that in Mongolia, they have reintroduced Przewalski's horses in what we will call reserves, right? So they are not yet without any fences. They are still protected by fences. Depending on the reserves, there will be some rangers not helping the horses, but making sure that no domesticate come in or that they don't jump the fence, right? So to ensure that they remain sort of isolated. And there are 3, as far as I remember, such reserves, and each does the preservation differently. Some are really actively intervening, and some leave the stuff going on by itself, right? There has been nasty winters in Mongolia over the last 20 years. One of the reserves lost 90% of their horses in one winter. But another reserve on the same winter managed to maintain all of the population. So you see, depending on what they do, they're at risk, a lot more at risk or not, to be preserved, right? So what our work has done is to sequence all of them and try to identify deleterious variants. So we have maps now of deleterious alleles in Przewalski's, and some people have started to clone Przewalski's horses, basically. And the reason for cloning is that, of course, clones are identical genetically, but they argue that if you clone females, you will give more chances for the population at the next generation to sire more horses, basically. There will be more females available to have kids, basically, to have offspring. And at the same time, those females that are identical genetically, they will create offspring that will be different genetically because of recombination. So by doing this, the cloning of females in priority, they expect that they could increase not just the census size of the population, which is important to fight against those nasty winters, but also the sort of reassortment of the genetic variation, the poor genetic variation that is preserved in the Przewalski's horses. So this is one way that in the US, companies, as well as the San Diego Zoo, companies in Texas, as well as the San Diego Zoo in California, have started to think about and elaborate on, right? There are other people, but this is, to the best of my knowledge, only being discussed, what I'm about to say, that they are trying to now propose models in which they will not only clone, but actually genome edit at the same time. right? So they would genome edit a number of targets. For example, perhaps some of the most deleterious variants that we could predict with conservation scores, for example. Typically, those variants that are always preserved across mammals, but are segregating as a mutation in the Przewalski's lineage. These will be the high targets for genome editing associated with cloning. But again, to the best of my knowledge, this has not been done officially, at least, or I haven't seen any paper, only people discussing this in conferences or meetings, basically.
We have a last question, maybe from Juliette Testas online.
Yes, thank you. Thank you for the talk. You partly responded to my question earlier, but I was wondering if the wild archaeological horses that you genotyped, which date back to before the third millennium, before the Common Era, also come from human settlements, and if so, if they were hunted by those population or.
Yeah, very good question. So the answer is depending on which, yes, or depending on which, no, right? So we have both cases. So we have, for example, I'm going to say, to give an example that is close to us here in France, Magdalenian people 15,000 years ago, we have settlements of those people. And there are a few horses that were found, for example, at the site of Étiole in the Marne Basin, dated to 15,000 years ago, 4 horses. They were hunted. We know this because that's a transitory site, and this is 15,000 years ago. Only the dog was domesticated at that stage. So clearly they were hunted horses. So clearly the answer is yes for that particular site. But we have also places still in France, for example, if I think about Cabreret, this is located in Dordogne. There is a trap, a natural trap, which is a sort of vertical cave, if you will, where animals that were running sometimes fell into that trap, right? And so there is piles and piles of skeletons of animals preserved in this particular natural trap. One is called Ig de Graal, for example. It's located like 2 kilometers away from the Pechmel cave in Dordogne. And this one, we have something like 20 horses we have sequenced. So clearly they were actually just wild. They just fell into a massive hole and they died at the bottom of the hole. And so we could radiocarbon date them from anywhere from, if I remember correctly, 13,000 years ago to more than 30,000 years ago. So you see, France, 2 cases, very different answers. One was a settlement, the other one was a natural trap. And in fact, we have a project that we haven't finished in which we compare systematically the natural traps versus the settlements to really see whether this is the same distribution of horse phenotypes that we could genetically predict, to see whether some particular types of horses were preferentially hunted by people. So we hope with this project that we will be able to learn not just about the horses, but in fact about the intention of past people. In other words, what kind of animals they were hunting. Was it random animals or one particular type only?
Thank you very much, Ludovic. Thank you very much. All obviously, we can talk about horses, like with great interest for long. Thanks again. Hope next time we will make it to Montpellier.
Yeah, I hope so as well. We are not so far, so it should be doable.
Yes. Thank you again, everybody. See you next week.
Bye-bye.